Okay, now for the experiment. So what they did in this experiment was they took bacterial auxotroph strain 1 and bacterial auxotroph strain 2. Remember, strain 1 was deficient in the ability to make methionine and biotin on their own, but they could make threonine, leucine, and thiamine. The second strain could make methionine and biotin, but not threonine, leucine, and thiamine, okay? So again, just to review, every organism, almost every organism, has the capability to take in protein from the environment and break it up into amino acids and take some of those amino acids and convert them to something else, all right? I'm backing up a little bit and talking about the physiology here because I think it's very important to understand the purpose behind these conversions. So in humans, we have 20 amino acids that our body uses. Our body makes some of them, but it cannot make the other ones. And those are the essential amino acids that we have to consume in our diet. Those would be actually biotin, methionine. There are a few others that you have to eat through your diet. And that would be easily, all those components come in easily when you eat animal sources of protein like chicken or beef. But you can also get them by combining vegetarian versions of protein like beans and rice or beans and corn. And so you get a complete protein dietarily by consuming the appropriate balance of different food components. So when we talk about proteins, I don't want you thinking about the chicken that you ate or the soy that you ate. I want you to think about chains of amino acids. And those chains of amino acids are individual building blocks strung together to make protein. And your body takes in the protein that you eat, breaks it down, sends it to your body through your bloodstream, and then your cells absorb the amino acids that they need to make new proteins. So it's a really interesting recycling mechanism that organisms have to take in nutrients and convert it to what your body needs to survive. That's what these bacteria are doing as well. They can make some of their amino acids, but some they can't. So what they did in this experiment was they took the first set of auxotrophs, grew them up in just what we call a basic medium, and then they put them on nutrient agar that lacked biotin and thiamine, okay? Over here, this group could make methionine and biotin, but it could not make thiamine. Okay, so over here we're lacking biotin. So this bacteria does not make biotin, number one. And number two, does not make thiamine. When you mix them together, these two groups, number one and number two, and you let them grow in the same test tube overnight, all these incubations were overnight, and you plate them on petri dishes that do not have biotin and thiamine, you got bacterial cells that grew. Over here, the cells did not grow because these cells needed thiamine, and this lacked thiamine, so you had no growth. Over here, in set number one, again, these agar dishes contain no biotin. These cells could not make biotin, so they died as well. But over here, all of a sudden, you have cells surviving. Why did that happen? So what ended up occurring to Lederberg and Tatum was that you started with auxotroph, cells that couldn't make this or this. You mixed them together and they could make it all. How did that happen? They weren't sure about what the actual transformation event was, but they knew that when you mix two auxotrophic strains together, you ended up generating what we call the prototrophs. They could become, they developed the ability to make everything. So that experiment was a seminal experiment showing that if you have two deficient strains, you mix them together, now they can survive no matter what. That's a little bit scary because what that means is all you needed to do in the beginning to kill this strain of E. coli was get rid of the methionine or biotin in the environment. Over here, to kill this strain of E. coli, all you had to do was get rid of threonine, leucine, or thiamine, okay? You just had to remove a food component and they died. But then you put these two gangs together, gang number 1 and gang number 2, or species number 1 and species number 2, and now the resulting species can handle anything, right? That's a little bit interesting. Actually, it's a lot interesting. So, a new experimenter came on the scene. His name was Bernard Davis. Now, Davis developed something called the Utube Experiment, and it's not the same thing as YouTube, what we're dealing with here. It's literally a tube in the shape of a U. And he asked the question, how do these two auxotrophic strains of bacteria actually give each other the ability to live in an environment lacking the nutrients they need? Do they actually have to touch, was his question. Because the idea is, here's auxotroph number 1. Let me see if I can do it this way. I'm going to say auxotroph number 1. And it has the ability to do some things. Okay. Here's auxotroph number 2. Can number 1 and number 2 swap information enough so that they both survive in the environment? So in the Utube experiment, what Davis did was he got literally a glass tube in the shape of a U, and he inserted a HEPA filter. This filter, a HEPA filter, has 0.2 micron holes in it. In other words, the holes are so little that bacteria can't go across. They can't touch each other, and they can't leak from one side to the other. But nutrients from one side can travel to the other side, but they can't touch each other. So if you grow these cells in a U-tube, one side contained a cotton stopper, the other side contained a stopper with a pressure suction thing. Don't worry, that it's a way to load these tubes easily. You don't have to worry about the cotton or the stopper for this to understand this experiment. If you take group number 1 and group number 2 and you grow them in a test tube that has a divider, but nutrients can travel back and forth, okay? Then you put them on the plates lacking methionine, threonine, leucine, and thiamine. Nothing grew. In other words, you change the condition from allowing them to mix together and touch each other to, and let's go back to this. They can mix together, touch each other, and they're all drinking the same Kool-Aid, okay? The same nutrient agar, or the nutrient medium, to the same nutrient medium, or, you know, bacteria Kool-Aid, and the bacteria are in the same environment, but they can't touch. And now you get no colonies, no growth. So what that means, or what Davis was able to refer from that, is that the cells need to touch each other to make this exchange of genetic information occur. Now, this group of information was developing at about the same time as the discovery of a process called conjugation. Conjugation is the process by which one bacteria passes a piece of DNA to another bacteria through direct cell-cell contact. Okay, so Davis's experiment was really important. The cells had the same environment. Okay, everything was the same, but they couldn't touch. So, in many but not all species, bacteria have the ability to touch each other and conjugate or exchange pieces of DNA. This was discovered by using a type of bacteria that carries a gene called F factor, or fertility factor. Now, don't confuse this again with fertilization. It was just called F factor because at the time they were still working out what is fertilization and what is conjugation. So they called this F factor, or fertility factor. So what you have in this case are cells that have F factor, and they're designated F+. Cells that do not have F factor are F negative. Now, where is F factor living? It turns out in a bacterial cell, so let's look at our bacterial cell again. Remember, the bacterial cell has a huge bacterial chromosome that snakes around. And bacterial cells can also get, it turns out, smaller pieces of DNA called plasmids. These are like the donut holes, okay? So if you think of a bacterial chromosome like a big piece of spaghetti, a plasmid is a circular small piece of DNA that is more like a donut. But this small piece of DNA it can be very powerful. It can confer resistance to drugs or metals. And when I say resistance to drugs, I mean like antibiotics. Plasmid DNA can also have genes on it that confer resistance to toxins that normally kill bacteria. And unfortunately, plasmids make bacteria pathogenic in some cases. Pathogenic means deadly. Okay? So what you're dealing with is a plasmid, a small piece of DNA that carries a few genes that make the cell kind of, you know, extremely dangerous to other organisms. Now, another term for F factor is an episome. This was the original word. Episome was the first word, and plasmid was the later terminology. An episome is a circular piece of DNA that lives outside of or extrachromosomally from the host's DNA. So this is the chromosome. This is extrachromosomal DNA. So F factor allows the process of conjugation to occur. F factor is, here's the F factor chromosome right here. It has this thing called ORI-T. ORI-T is where DNA synthesis starts. When we get into the molecular biology, we're going to start talking about initiation of DNA synthesis. It always happens at the origin. And here its name is just ORI-T. And this whole plasmid or episome is called F factor. When you look at each one of these little words like TRAP-A, TRA-A, not TRAP, TRA-V, TRA-C, each one of these are genes that play a role in transferring a small piece of DNA, the actual F factor, from one cell to the next. So here are two bacteria. This cell has F factor. I'm trying to get different colors to come up, guys. Okay, here's the regular cell. Oops, it didn't do it. I'm trying to change colors here. Still learning to use my pen, folks. Here's the regular chromosome. The two cells have the regular chromosome, but this one cell has F factor. So what has to happen is F factor has to go from this cell to this cell, okay? And when that happens, that transfer, that is the process of conjugation. And each one of these genes here plays a role in that. This TRA-D is what we call coupling protein. TRA-I, or sometimes people call it TRA-1, but it's an I, is the gene for a protein called relaxase. It causes the plasmid DNA to relax and kind of, instead of be real tight, be real smooth. There is a transporter protein that literally links the two cells, okay? So this cell is going to reach out and form what we call the conjugation pilus. The conjugation pilus is formed by the pilin protein. And then the transporter, okay, which is also in green here, I'll make it a lighter green, gets this moving across to here. Now, there's a better picture coming up. All right, so conjugation occurs when the sex pilus, or they also call it the F pili. I mean, you know, again, this isn't sex. It's only made by F plus strains of cells, okay? And it makes the pilus, or the tunnel through which the DNA travels from F plus to F minus. So if we come back here and we label our cells, okay, this would be F plus, and this would be F minus. Once the conjugation has started, the conjugation event usually finishes up. The conjugation bridge or pilus is formed between the two cells. Okay, so the bridge is the same thing as the pilus. Okay, and the donor cell is the F plus cell. The recipient cell is the F negative cell. And so here's a picture showing the whole process. The donor, F plus, the recipient, F minus. Here is the conjugation pilus right here, and the exporter is a special protein that allows the plasmid to go from one side to the other. You'll see that a piece of the relaxed chromosome peels off, enters the other cell, and voila,